Good evening, everyone. Welcome to the session, uh, which is fifth week session of Plants and Bioprocessing live session. Um, we will discuss few questions here, and if you have any doubts on the lecture series, uh, which is for week 5, we can discuss here. Um, so before going into the lecture, can anybody tell which are the lectures was covered in this week, which is week 5? What are the lectures being covered in this session? Last week we were halfway through optimization strategies. So week 5 was what? It is optimization strategies and your immobilization, right? Okay, so we'll go through the questions plus biotransformation. Okay, thank you, Susan. We'll go through the questions and then keep on discussing about the related topics. So the first question is, casein hydrolysate and peptone are used as dash sources in plant tissue culture medium. The answer is carbon or nitrogen. What would be the answer? Yes, the answer is B, which is nitrogen. So the first question is itself about plant tissue culture medium. So can you guys give me answers on what are the sources which will be present, or the nutrients which will be present in any plant tissue culture media? First, first we will discuss them, and then we will go back to the different types of media available. So what do you think are the major sources of energy or nutrients needed for plants for in vitro culturing, which would be present in the plant tissue culture medium which we are preparing? So the answer here, you told that it is, the answer for this question is nitrogen. So obviously nitrogen is there. What else will be there? Nitrogen sources will be there. What else will be there? C in mineral growth regulators, yes, carbon, nitrogen. The carbon source will be there, nitrogen source will be there. Minerals in the sense, what are the minerals? Growth regulators, yes, absolutely. Growth regulators will be there. Growth regulators are phytohormones, we can call them. Okay, micronutrients. And what else will be there in a plant tissue culture media? Micronutrients, macronutrients. Why are they called micro and macronutrients? Any clue? Any idea? What are micronutrients? What are macronutrients? Why they are called micro? Why some nutrients? Amount, yes. The amount they are being required by the plant, that's why they are classified as micro and macro. The macronutrients, they will be required in uh, greater quantity compared to your micronutrients. So what are the examples of macronutrients? Any two examples can you give for macro and micro? No idea. Any, so you guys told minerals, right? So it will come under macro and micronutrients. So Yes. Macro CH. No, 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 no. CH is entirely different. Micro in the sense, carbon is separate source, nitrogen is separate source, oxygen we are not going to give, growth regulators we will be giving. So these both generally include your minerals and salts actually. Um, Say for example, copper, magnesium, potassium, phosphorus. These are required in higher quantities compared to your cobalt, manganese. So these are micronutrients. Micronutrients are comparatively added in lesser quantity. Your calcium also comes into picture. So these are examples I'm giving. So you have heard about for natural plants which we cultivate in fields and all, we add NPK fertilizer, right? So why we do add that NPK fertilizer? Urea we add. What are those things? In paddy field, whatever field we have, generally we are cultivating, whatever we are cultivating, we use NPK fertilizers and urea apart from what you call fungicides and weedicides, herbicides and all. We use these things. Why we use that? N stands for your nitrogen, P for phosphorus, K for potassium. So these are macronutrients along with nitrogen is being added to the soil to aid in plant growth. We are giving in the form of fertilizer. So similarly, for any plant tissue culture media, there will be a nitrogen source. Nitrogen source can be in the form of water. What one in this question it is given casein hydrolysate, casein, peptone, and other sources, other major sources of nitrogen for plant tissue culture medium is what? Any idea? You guys would have gone through the slides, lesson lectures, na? Amino acids, amino acids, ammonia. More than amino acids, ammonia we can call them as a except your inositol, others won't be considered as amino acid. So like as a source of nitrogen then. So carbon, what are the carbon sources? You can give glucose, you can give fructose, maltose, whatever, but majorly used is sucrose. Sucrose is the major source which is provided in the plant tissue culture media. Okay, so the answer for this question is casein hydrolysate and peptone are used as nitrogen sources in the plant tissue culture medium. So, for example, last or week 3 we were talking about uh, optimizing the media components for secondary metabolic production or primary metabolic production. Those. So, say for example, my compound of interest is produced by the cells during the secondary metabolism. So I have to harvest the cells or I have to let them grow until the early stage of stationary phase. So if I need more cells during the stationary phase, means what I have to do. Because during you all know that say during stationary phase growth stops. So whatever growth which happened during your lag and log phase only will be maintained in your stationary phase, right? So generally, certain ratio is there where your nitrogen ratio. Have you like? Do you guys remember about when professor was talking in the slides about this ammonia ratio? Ammonium, yes, ammonium nitrate ratio. So, what was the reason about increasing or decreasing the ammonium or nitrate ratio, ammonium to nitrate ratio? So you guys have, I think it was in the macro and micronutrients when professor was talking about macro and micronutrients. So the use of nitrogen, phosphorus, and ammonia, potassium, everything, it will suppress your rapid cell division. So when cell division is being suppressed, what will happen? It means that it suppresses growth. So when your growth is being suppressed, what will happen? See, you are changing the media composition in such a way that if you increase your nitrogen, phosphorus, and ammonia content, what will happen? And if you reduce it, what will happen? Based on this, you can get more secondary metabolite. That's why I'm asking. Yes, Sashank is right. If you alter the nitrate and ammonium ratio, whether we have to increase this or decrease this, which will aid in secondary metabolic production. Any idea? Shashank was giving the answer. He said that the secondary metabolic production will be impacted. Okay, Komalavalli is telling that if we increase the nitrate to potassium, like ammonium to nitrate ratio, secondary metabolic production will be high. Okay. Any other contradicting answers? No answers. Anybody think Komala's answer is correct? So we are talking about ammonium to nitrate ratio, nitrate to ammonia ratio. The ammonium to nitrate are no decreasing might increase production, I guess. Okay, both contradicting answers are there. So why do generally ammonia and nitrate? They both are nitrogen source only, na? What is so different about ammonium nitrate ratio? Ammonium aids for growth, okay. So they both are nitrogen sources. Only the way they are getting assimilated or used by the plant is going to be different, right? So one may be needing some breaking down into something else, and nitrate is easily converted into nitrogen source. Do you think that that will have a cause in this? So I will give you a clue. So if you increase the ammonia to nitrate ratio, there will be decrease in the growth of plant cells. So you guys tell me with whether we have to increase or decrease for getting higher secondary metabolic production. if you increase the ratio of ammonia to nitrogen in the nutrient media, it will cause decrease in growth. So do you think whether to get an increased secondary metabolic production, do we have to increase or decrease the ammonia to nitrate ratio?
I'm decreasing.
Yes, we have to decrease it. So it will suppress the growth, and more metabolites inside the plant cells will be directed towards secondary metabolic production. We'll go to the next question. So the next question is about plaquebermidazine. The the factor dash assumption in plaquebermidazine states that only a few factors are responsible for variation in the data. So what would be the answer for this? This is one of the assumption of plaquebermidazine. So the factor dash assumption in plaquebermidazine states that only a few factors are responsible for variation in the data. So what is the answer for this question? Any idea? The factor dash assumption in plaquebermidazine states that only a few factors are responsible for variation in the data. Okay, first, so do you guys know what is plaquebermidazine? Have you gone through the slides, lecture slides, and you guys were able to understand plaquebermidazine? Okay, Sashank knows. Then Sashank, what is the answer for this question? The factor dash assumption. What is the assumption? This is one of the assumption of plaquebermidazine. If you find under like able to recall the assumption, you will be able to answer this question. Parameters are independent. Okay, the factor parameters are independent. Okay. See, read through the next line, which states that only a few factors are responsible for variation in the data. Should I tell the answer, or you guys will take guess? So the answer is the exact. Okay, okay, it's okay, Shashank. Sparsity. The factor sparsity assumption. That means that there are only few factors among the various factors you have selected will be responsible for variation in the data. That is the assumption. Okay, now who and all understood plaquebermidazine will now help me in putting a plaquebermidazine table for 7 factors. Let's say for 7 factors, somebody who already understood plaquebermidazine will help me in designing the table. Okay, before that, why we do plaquebermidazine? Why suddenly some design and all coming into picture here in plant cell bioprocessing? Nobody understood or nobody wants to talk to avoid OF80. What do you mean by OF80? To avoid OFAT, one factor at a time. Yes. To avoid doing multiple runs, one factor at a time. So for those who don't understand one factor at a time, I will tell about it. So say for example, I have carbon, nitrogen, and mm, carbon, nitrogen, and some what do you call some phytohormones, some phytohormone concentration. So these are my factors. So let's give first factor, second factor, third factor. So if I vary one and keep one, if I keep this concentration of C, which is say for example sucrose we took, if I fix this concentration as 1 gram of carbon source, and nitrogen if I vary to 2, and phytohormone also I will keep it as a fixed one like 1 gram. I will do one trial with this, first trial. Then I will do 1 gram, I will vary this, keep this as fixed, and then do increase it or decrease it, and do like that. So I will do second trial, and then I will it keeps on going. You will fix one factor at a time or vary these two. See, factor is components we can keep. So if I fix my carbon source and then vary my nitrogen and phytohormones, that means it is one factor at a time. You are fixing one factor and varying the others. And if you keep on doing this, it will take a lot of time, your time, and it will take a lot of your energy, and even your experimental runs will take a lot of time. So what people have come up with is to avoid this doing one single factor at a time, they have tried doing multiple factors at a single time. So all these factors will be done at a single time. You can fix this at one point, vary this. You will fix this at one point and vary this. Like that, it keeps on going. That is what is Plackett-Bowman design. So if you keep on doing one factor at a time, it takes a lot of time. So we will go with Plackett-Bowman design. Okay, this is for what? Screening or selection? Screening, yes, because if your media has, say for example, in your literature, there are multiple media combinations or multiple literature available. So in each literature, they have told that one person is telling that sucrose is the best carbon source, one is telling glucose is the best carbon source, the other one is telling maltose, the other one is telling fructose. So different different factors are coming into existence here. So if you keep on doing one factor at a time, what you will do, you will keep 30 grams like this, fix it, vary this, fix it, vary this. Then you will find, oh, 30 grams of sucrose is working out for me. Like that, you will keep on varying things and finding out which one is giving you good result. Good result here in the sense, good biomass growth or higher biomass growth or higher metabolic production. That is your ultimate aim. So, in case of this, Plackett-Bowman design has been introduced where they assume that the factor sparsity is there, and which by mean it means that only a few factors are responsible for variation in the data. So according to the Plackett-Bowman design, they have taken the factor sparsity assignment assumption, where it means that maybe not all the factors which you have taken are impacting the result which you are anticipating to get. So that is the assumption which is stated here. Okay. So now give me, so those who, okay, Sashank is answering. So I'm understanding that he remembers and understood the Placket-Bowman design and read about it. So what about others? We are still in Placket-Bowman design. There is a response surface methodology and all coming at the back. Everybody understood, if if somebody doesn't understood any about Placket-Bowman design, raise your hands. We will discuss in the next session with a case study. Everybody understood, huh? Okay, then nobody is answering then except Sashank. Should I ask questions? Guys, you really understood plaque-bomb design? Okay then. Okay, then let's leave plaque-bomb design. We will go to response of his methodology. Na? At the time, I will see whoever is answering. Okay, we will go to the next session. We will come to this plaque-bomb design at the end. We will see whoever is. I will ask some questions, and whoever is answering, we'll see. If you have doubts in plaque permit design, na, ask me. I'm here to solve those doubts only. Okay. So dash method is used to determine the optimum response for a specific range of variable conditions. Which method is used to determine? Yeah, response surface. So here are some of the, what you call, if your answer is response surface, there are two response surface methodologies being kept here, box bending and central composite design. Which among these are the method used to determine the optimum response for a specific range of variable conditions? The answer is B and C. You are right, Shashank. Both of them are responsiveness methodology only, which is box Benkin and central composite design. So, what is the difference between both? Nobody is answering for placket Bowman itself. So, any idea what is box Benkin design and central composite design? No idea about them. Okay. So in your placket domain, what you guys will do, you will screen out the important factors which are affecting your experiment, or yeah, you can call it as experiment. So say for example, you are in the initial stages of your research and you are fixing the placket Bowman design where you give higher and lower values only, like one high value you will give and one low value. Say for example, if it is a carbon source, high and low, nitrogen source, high and low, and your micronutrients, high and low. Like this, you will keep on doing this. And say this is three factor, right? Carbon, nitrogen, macro is there, phytohormone is there, like that. So from this you will identify the most significant one, means which one is positively impacting your design and negatively impacting your design. So positive, positively impacting your design in the sense, say for example, carbon is giving you which the design which have plus sign of carbon and minus sign of negative, like nitrogen, and minus of micronutrients, means this is giving you better growth, means nitrate and micronutrients are negatively impacting the growth of your cells. That is the idea. So you will first screen out that which are the important uh, factors which are positively impacting your cells to grow or production. So from this placket Berman, I might have come to a conclusion that okay, my carbon source is important, my nitrogen is important, and my micro is important. Leave the others. Phytohormones are needed, but others like I'm giving a entire picture here, but generally it will be like in the nitrogen, how many different sources are there? Na? ammonia people will keep, and other nitrate form they will keep, ammonium nitrate they will keep, like the different forms are there. So first we will find out the important factor, and then we will go to response surface methodology method, which is either we go with CCD, central composite design, or box bucket design, and full factorial design is also there. So what we will do, we will instead of in your placket Bowman design, you went with high and low value of a particular factor, but here you will give different data points. Say for example, so in case of your CCD, there are three different points we call. One is axial points, another one is your center points, the other one is axial, center, and star points. So, what do I mean by points? The range which I'm going to take. So, say for example, my objective of my study now is to optimize my carbon source, out of which I'm taken glucose. Say for example, glucose I have taken. So this glucose different range, high like lower range is 2 grams and up to 100 grams. I need to optimize this. So I cannot do 2 gram I will put and wait for the result, 3 gram I will put and wait for the result. I can't do that, right? So what I will do, this is my working range. In this I will take a center among this center point. Say for example we will take a 50. So that will be my center point, 50 grams. And my actual points will be where it will be. They will be at the, say it is like a cubic design. So this is my center point. So this is my center point. Why generally center points are kept? Any idea?
To compare and then optimizing.
To compare and optimize, or we can call to reduce any error. So if you keep one extreme and the other extreme and there is no center one point, means you don't know, like, to just to avoid any errors happening in your design, we generally keep center points. So center points, people keep n number of center points. They will, while they are designing their end, they will keep 2 times or 3 times they will keep center point, so that their error will be minimized. So center point is at the center, and axial points where they will be at the axis, right? So axial points are the, say for example, if I had taken 2 gram to 100 gram, then my axial point is what? My range is 2 gram to 100 gram. And I have kept an center point of 50. Okay. So what would be my axial point? 200. Yes, right. 200 is my axial points. So 200, these are my actual points. We are discussing here only one factor. So, so don't worry about why I have put many points. Like, to make you understand how this, in the form of Q only, they generally represent this CCD and BBD, which is box blanket design. So now center point is here, axial point is at the corners on each side. And where will be the star point? Star points are kept at a distance of alpha. Alpha is nothing but it is a particular distance, at particular distance, specific distance. You can go and read. I will check and whether if it is. Do you guys still don't have access to the drive link where you can see the PPTs, like the at least the lecture PPTs, which uh,
Yes, ma'am. We are getting
you
it.
are getting, right? Okay, so I will share them with you material for the CCD and BBD. So these star points will be at an equivalent distance from the axial point. The distance would be alpha. From axial point, alpha distance away from your axial point, star points can be placed. This alpha depends on number of factors you have chosen. It varies. Okay. I will share you. Remind me once I will share with you about the alpha and everything will be given. There is a formula for calculating alpha depending on the number of factors you have chosen. Now I have selected only one factor to help you visualize things. So I'm not giving the value of alpha here. So alpha is nothing but your star point is nothing but at a fixed value of alpha distance or alpha value away from the axial point, your star point will be placed. The distance between your axial point and star point will be star point will be alpha. Okay. If it is in the minus axis, it is minus alpha. In the plus axis, is plus alpha. So that's all. So your alpha value, say for example, if I give my alpha value to be 1.21 something. So minus alpha plus alpha. Then your one axial point is 2 means we will go minus alpha side. 2 minus 1.21. What is the answer? That will be your axial point, like star point. Then your 2 gram will come, which is your axial point. Then your center point, which is your 50. Then your 100, which is another axial point. Then your plus alpha, which is this side, 100 plus 1.21, which is 101.21. So these are the 5 points which will be needed for your placing your CCD design. Sangamitra, 51 n plus 1. What do you mean by 51 n plus 1, Sangamitra?
Point?
You have written 51 n plus 1 in the chat box.
Yeah, it's is it a star point? Can we take n as a factor and n plus 1 is 1 point which is up, so it will be the star point will be 51?
Uh, for center points, you don't take actually.
Okay.
Yeah, it is actually alpha distance away from your axial point.
Okay.
Yeah, that's right. So if your alpha value you fix it as 1.20 or something, if your range is 2 and 100, then this alpha is this. So both in minus axis and plus axis we will see. So minus alpha you have to do with this and plus alpha you have to do with this. I think if I give you a learning material that will be very easy for you guys to understand more than trying to make you understand here in video call. Okay, so this is your what is that? CCD, Central Composite Design. So there are few differences between your box Benkin design and CCD design. So in your sorry, in your CCD you can go up to certain levels or you can visualize up to certain factors. But in case of BBD, you cannot visualize the design. If it is less than 4 factors, you cannot do that. If it has more than 4 factors, you can go with box Benkin design. And also, box Benkin design will can help you have fewer runs compared to your CCD. So in case of CCD, say for example, runs, if I mean by runs means 30 different combinations. All these combination you have to experimentally try. Then the experimental data you will put it back to the design, check it. It will give a predicted response by fitting all your responses in a quadratic equation. The predicted response and your experimental response will help you understand what is the uh, range, or what is the say for example, if you are trying to identify the concentration of glucose which gives you maximum growth or metabolite content, so that will give you which range you have to, not which range, which particular concentration you have to take, and while taking that concentration how much is your response, which is either growth or metabolic production. So this will be easily found by using your RSM, and it also helps you understand interaction between the factors. So Placket-Bowman doesn't have any uh, in it's not built in way to identify interaction between the factors. Say for example, if I'm taking ammonia and nitrate as one factor is ammonia and the other factor is nitrate, Placket-Bowman won't give you whether both these ammonia and nitrate are interacting among each other and giving that response. You cannot see that whether any interaction is there between that, but you can visualize them in your CCD and BVD. That is the difference. Okay. I'll try to put the learning material for response surface methodology and placket permanent design for your reference in the drive link. So with that being said, we will go to the next question, which is failure in secondary metabolite enhancement upon addition of precursor in the medium can be due to its diversion into alternative pathways. Do you think the statement is true or false? True.
True.
Okay. So how did you guys come to conclusion that this answer is true?
That may be due to that feedback mechanisms and all the addition of precursors.
Okay. So you have added precursor into the media. So your expected outcome is that you have added your precursor in excess in the media, and as you have added your precursor already in the media, the cells will think that it is already overly abundantly present in the cell system. So they will start diverting the entire precursor, like kind of transforming them into your end product. But it is not happening. That is what the question is about, right? So while choosing the precursor itself, what and all you have to consider? If you choose a precursor, say for example, so this answer is A. So if you choose a precursor which is A, and this precursor is being converted in few 3-4 steps or 5 n number of steps into your end product E. Let's keep it like this. So while selecting a precursor A, what are the criteria you can think of to avoid this situation? Limited uptake. Okay, if you limit the uptake, one way it is right, Shashank. If you limit its uptake, of course, it will concentrate more on the what you call mostly necessary way it will go and then it will produce it. Okay, we can take, if we limit the uptake. So if you are going to limit the uptake means you are going to provide less in the media, right? If you do so, there may be a unnecessary diversion can be avoided. That is there. Okay. What else? So while choosing the precursor, say for example, instead of using this precursor, you could have gone to a very specific precursor. Say for example, there is C, and the Ca can be diverted into something called G and something called H, something called I, everything. So what you could have done? You could have selected this one instead of going for this, which you know that it will get diverted into these many places, right? Do you guys agree? Yes, so what you can do, like if you are aware of the pathway, which most of the time we are not aware of the pathway, because the pathway in which the particular metabolite is being made is most of the time is not available. So in those cases we cannot help. So but if the pathway is available, you can go with very what you call, which doesn't have any other diversion routes, so that your entire precursor you are getting, you are adding into the system will be sent back to your end product. For this, you guys have any of you studied metabolic engineering or fermentation process, a book by Stanberry, I guess? Have you guys gone through any books like that? Have you gone? Okay, Fauzi was talking about feedback mechanism, feedback inhibition, right? So everybody know about feedback inhibition in any system? So there are multiple ways you can increase secondary metabolites. So one such way is addition of your precursor, addition of elicitors, everything, and you can at a certain extreme you can go with metabolic engineering to modify the pathways. But the problem in here is that you need to know the pathway. So the answer could be would be like Shashank said, you can limit the uptake by providing less in the media so that it won't concentrate too much on the other side pathways which are of not, they don't have much importance, means they can nullify them, and they can go with the next metabolites, or you can select another suitable precursor which doesn't have more metabolically energy, like metabolic energy load on it, so that it can divert its pathway directly into the end product which you need. Okay, coming to the next question. Dash, when added in the medium, can stimulate stress or defense related response in plant cells, resulting in enhanced production of secondary metabolites. Elicitors, right, exactly. So elicitors, when they are added in the medium, they can stimulate stress or defense related response, which will further aid in the enhanced production of secondary metabolites. Now, I think in last week, we were discussing about elicitor, precursor, everything. So now, tell me the difference between precursor and elicitor. I think last week also we had the same discussion about elicitors and precursors. What are elicitors and what are precursors?
Elicitors will induce the production of this
Okay,
metabolism.
But precursor also does the same, na?
The precursors means the substrates that is uh, involved. The intermediates,
Okay, elicitors are intermediates or precursors are intermediates?
the precursors,
Okay, precursors are intermediates. Then elicitors are what? Okay, elicitors they stimulate defense. Precursor they are already present in the pathway. Intermediate. Okay, Sashank has given one answer. Fauzia was also telling the same. So precursor is just only which is present already present in the pathway, or it can be endogenous or exogenous also, na? It can be either derived from the or it is being already produced in the same micro plant system or not, or exogenous also, but it should directly enhance product formation. Yes. So likewise in your elicitors, precursors also there are endogenous and exogenous precursors. Okay. Do you guys remember natural intermediates and obligate intermediates? What are those? Natural intermediates and obligate intermediates. No idea. Natural intermediates are what? The name itself has it. These are produced from the organism. These will be produced by the organism of interest itself, which is here plant. So obligate is what? What is obligate intermediate? Externally supplied, it can be used to produce product. Okay, partially it is right. Okay, this week's small challenge is to go and read about obligate and natural intermediates. Okay, we will come back to that next week. I will ask. I have to remember to ask. Okay, Sasank has partially answered it correctly. So you guys go through and give me the answer next week when we are meeting for week 6 discussion. So coming to the elicitors. So now come to the elicitor. Elicitors example, can you guys give me one or two examples? I won't ask more questions, please. Few examples. Likewise in your precursors. Okay, calcium. Okay. There are various forms of elicitors, right? Like forms of elicitors are what generally they are anything which gives stress to your plant system is elicitor considered, right? So even your elicitor can be given as like biotic elicitors and abiotic elicitors. What is biotic and abiotic? Anything which is produced by a living organism is biotic, and anything which is non produced by a non-living thing is a abiotic, right? So can you give some example for biotic elicitor and non-biotic elicitors? What will come in biotic elicitor? Biotic is pathogens. Yes, when a pathogen is trying to give entry to your or get into your plant system, they may produce some compounds which may cause defense, which will cause defense mechanism to start in your plant system that indirectly or directly causes a improved production of your secondary metabolites which will act on the particular organism, right? So like Sashank is answering abiotic, yes, temperature, light, salt, UV, sonication, all those things will come with abiotic. So temperature, if you give harsher temperature, like say for example the plants will withstand only 25 degrees Celsius and you're giving 30, means what will happen? They will undergo stress, right? It is that is not the temperature they are accustomed to. So this will in turn cause stress response in the system and they will start producing the secondary metabolites to contract the effect of derogatory effect of your temperature. They will start producing secondary metabolites. Same goes with the presence of salts and like say example, if your plant cells are they need only a light intensity of 1000 lux and you are giving them 3000 lux, what will happen? They will try to survive even under the stressful conditions so that they will start producing secondary metabolites which may or may not help them in contradicting the like contracting the uh, negative effects of the light on the cells. So this way your elixir helps in increasing the secondary metabolic content. So when exactly you need to add elixir to your cells, plant cell systems? Say for example you have suspension cell culture. So what is the time point where you plan to add your elixir? I think last week also I had asked the same question. When exactly we have to add any elixir? You can add in the start of the culture itself, like while you're starting initiation. Which phase you will add? Lag phase, log phase, or stationary phase? When you will add? End of log. Yes, end of log or early stationary, you can add it. Good. So the answer here is elicited. Okay. So the sixth question is, I think only one more question is there. So the method of immobilization where binding occurs due to natural affinity is called dash. What would be the answer for this question?
The adhesion,
Adhesion,
agglutination,
agglutination,
then adsorption, I think.
adsorption. Yes, you are right, Fauzia. So the others won't happen naturally, or they don't have any natural affinity going on there. Yes, Shashank, you are right. B, E, and G. So adsorption means what? What is adsorption?
The non-covalent or the weak forces of attraction.
Okay, their weak forces of attraction will be there between the adsorbent and the absorbing particle surface. Okay, so what will happen? So they will get settled on the surface of your adsorbent, right? So that is one way of doing it, adsorption. So what do you mean by adhesion?
The attachment between.
It is a general way of sticking to each other, we can call. Naturally, a compound is sticky in nature, means what will happen? Anything which goes nearby will get stick to it. So that is also coming under natural affinity only, right? So why the answer agglutination came? So what is agglutination? Absorption is naturally due to the weak forces of attraction between the adsorbent and the adsorbate. Absorption is happening. Adhesion is anything which has a sticky nature can have affinity towards something, so something gets stick to each other. Surface phenomena with surface energies so involved. Yes, yes, Joey, it is clump formation. Agglutination is formation of clumps due to that also due to the natural affinity happening between the two things. Your gel and what other? Your gelling polymer and your cells, for example. So these are natural affinity containing agents. So why cross-linking, microencapsulation, covalent coupling, entrapment, why they didn't come under natural affinity? Because you are making them do that, right? What you will do in microencapsulation? What is microencapsulation? What is microencapsulation? What will happen in microencapsulation? Different pore size framework encapsulates cells. Okay. We can take this as the example can be calcium alginate method. Can we consider calcium alginate beads, sodium alginate beads as an example for
Yes,
microencapsulation?
yes, yes, yes.
Yes.
Yes, of
So
course.
then what do you mean by entrapment? Example gel entrapment. Okay, in a gel, everything, all the particles or your cells are getting entrapped in it. They are not formed into beads, but they are getting just like a bed of things. Say, for example, like this, they are getting entrapped inside. Okay. So, what is cross linking then? These are all chemical process, right? Right. So, you are making your with the use of specific reagents and with the help of your covalent or other bonds you make or yeah connection via chemical agents. Yes, you will base these are based on basically your gelling polymers and the chemical reagents will be used to cause a cross linking bridge kind of things inside so the your cells are getting entrapped inside only. So okay, so you are learning about immobilization here, and why do you first of all go for immobilization? What is the purpose of doing immobilization? Long term use of cells, yes, long-term usage of your cells and long-term use of any other in case of any other systems, say for example enzymes and all they use they entrap the enzymes generally inside so that they use the enzymes for so long. Same goes for your plant cells also, and you can um, use the process continuously. No need to grow your cells again and again. If your cells are already entrapped, you are just passing things inside and getting things out. So you can use it for biotransformation also. So what does biotransformation do actually? What is biotransformation? What is biotransformation? Conversion of chemical forms. Okay. So, say for example, a compound is going in as A and it is metabolized by the plant systems and it is being converted to an end product of your needs. So instead of isolating some enzymes or something, purifying or even in microbial cell system, they entrap the entire cell and they use it to transform one compound to another, because this will reduce your production cost a lot, because you cannot fully purify the enzyme and use that enzyme which is converting this substrate into product. So you can use plant cell systems as such, entrapped inside or encapsulated inside beads, pass your substrate of interest and you will get other than your product of interest. Yes. So what do you think that this immobilization might have a disadvantage of? It has many advantages, like you can run the system continuously, you can reduce the problems of aggregation, foaming everything, because the cells are not in contact with each other and the cells are being separated from the media and kept aside. And you can even have higher biomass levels compared to normal way of growing the cells, because if you do high biomass density in a reactor, what will happen? There comes the big problem of mass transfer limitations, which include problems associated with aggregation and foaming, which you can avoid here because the cells are already entrapped or encapsulated. They are not in contact with each other, so the problem associated with aggregation will go, foaming will not be there. But among all these advantages, what do you think that there are disadvantages of the systems, which is plant cells being immobilized? What are the disadvantages? Product release, you are right. So the release of product depends on the permeability of your membrane. So if that is not going to work up properly, then your product, even though they are released by the system, may not reach your broth. So you cannot purify them further. So membrane permeability is a problem there. What else is the problem? So if some, say for example, some compounds may degrade if they are coming in contact with some other things. So in those kind of things which we don't know about or we have no idea about, or say for example 100 mg or 100 kg of biomass you are using, 100 kg say for example, out of it your aim is to harvest at least 1 kg of your metabolite. So what will happen? It might have produced 1 kg, but what you get after separating it out, everything will be around, if it is around 600 700 mgs means you don't know that there might be some degradation going on due to the presence of immobilization membrane covering your system. And like Sashank pointed out that your permeability is not there, so product is not getting released properly out. Anything could have happened. So these are some of the disadvantages, and there is also a fear that it might reduce the biosynthetic capacity of the cells itself, because you are isolating them in an environment. So in some cases of cells, maybe they need what close contact with other cells so that they produce uh, the metabolite of our interest in a better way, or higher production can be anticipated. So these are some of the disadvantages which might occur due to the immobilization of the plant cell systems. So the last question is, culture age cannot affect biotransformation. The answer for this question, true or false? False. Yes. So the age of the culture matters. Age of the culture is like inoculum age is very important. So if the cells are not in the phase you are looking for, then the biotransformation may not happen, right? The answer to this question is false. So I think we have pretty much covered the topics and discussed few. If you have any doubts, you can ask me here. If you don't have any doubts, you can leave the call. If you have any doubts, you can ask. Do you guys have any doubts?
उसको तो कह दो भी उसको। हाँ रिक्शे पे चली जाना, रिक्शे पे चली जाना, साथ मरवा को भी ले लेना।
मरवा में स्कूल जाना है, घर पे तो मैं नहीं,
वो पहले ही छुट्टी करन दे तैयार बैठी।
उसके पेपर होने वाली है।
रिक्शे पे तुम दोनों को अकेली को कोई जाने देगा?
नहीं हमें जाए, मामू स्पीक बाइक पे नहीं ला सकते।
वो कहती है अब्बू ने नहीं करने देना। उन्होंने इतना कोऑपरेट मेरे साथ। भाई का दोस्त तुम्हें ड्रॉप कर देंगे 11:12 तुम दोनों को। सुबह जाते हैं ना वो, या शाम हमारे से रिक्शे पे चला चले या कोई ऐसे। तो शाम स्कूल जाता है भाई का दोस्त, सुबह जाते हैं ना दफ्तर 7 बजे, 7 बजे जाते हैं ना, 8 साढ़े 8। उस वक्त तुम दोनों कल रेडी हो जाना, वो तुम्हें ड्रॉप कर देंगे, वहाँ से रिक्शा पकड़ के आ जाना। चलो ऐसा करते हैं कि ना महल ना उसी महल वाले में सीखे मेरा ऐसा करते हैं कि अगर ना मैं यहाँ से रात को रुक जाऊँ ना सुबह गया तो दूसरा मैं फतेह शहर छोड़ दें। तुम रुक जाओ ना, वहाँ से तुम रुक जाओ। हाँ, अगर हाँ, और कल चली जाओ जाके स्कूल लिविंग सर्टिफिकेट दोनों का ले आओ। कल फिर
Uzma, can you mute yourself?
फिर मंडे को भी इसी तरह कर लेना ना। मंडे को फिर दोनों जो है वो कदूस भाई आप लोगों को ओपन यूनिवर्सिटी के दफ्तर ड्रॉप कर देंगे, इनको पता है, वहाँ अपना काम कंप्लीट करके और उसी तरह फिर वापस आ जाएंगे। ऐसे रख दो, पहले ये काम तो करके आओ ना, वो बाद में कर लेना। पहले ये कल जाके भाई का दूसरा
Thank
ड्रॉप
you
कर
for
देंगे,
joining. We are at
रिक्शा
the
लेके
end
आ
of the
जाना।
session.
इसी तरह फिर
So we
उधर
will
से
meet
जो भी
next
इसने
week
लेना है
to
बेफॉर्म,
discuss about the
वो
week 6
लेके फिर
session of
फतेह शहर
plant
चली जाना तुम
bioprocessing
दोनों।
live. Thank
और
you for
वहाँ से जो
joining.
है ओपन यूनिवर्सिटी के तो ऑफिस अब्बू भी ले जाएंगे, फतेह शहर से हमारा भी अब्बू ले जाएंगे।